of YCIS Satara in second day panel discussion on the platform of YCIS Hackathon 2021. I welcome Honorable Principal Dr. B.T. Jadhav sir, eminent panelist Honorable Dr. Samir Naik sir, Assistant General Manager in Serum Institute of India Private Limited. The second immediate panelist Honorable Dr. Arun Kharat sir, Professor in Microbiology, School of Life Sciences, JNU, New Delhi. Mr. S.K. Shinde sir, Coordinator of YCIS Hackathon, all the coordinators, faculties and students. The topic of the today's panel discussion is drug and vaccine development. This is a topic in which we all are deeply interested because of current COVID-19 situation. And we are very honored to have eminent panelists Dr. Samir Naik sir and Dr. Arun Kharat sir. I would like to request Dr. V.M. Ghorpade sir to introduce first panelist Dr. Samir Naik sir. Over to you, sir.
Okay. I extend the warm welcome to all in Hackthon 2021. I find immense pleasure to introduce the Honorable Dr. Samir Naik, when he had been working for past 15 years in the Serum Institute of India. He is now working as Assistant General Manager, Quality Control. He is QC in charge, the quality control in charge for the viral vaccine, including Covishield, rotavirus, RSV vaccine. He had 15 years experience in the biopharmaceutical industries and the vaccine development technology, transfer, analyst, and the product life cycle management. On his cadet, there is 2 international awards. One is Deals of Distinction Award from USA and the Canada. And the second one is the Humanity Award from United States Trade Organization. He published many research articles in international journals. We are really thankful to him out of his busy schedule to attend this function. With this short introduction, I conclude and I hand over the charge to Manisha Shinde. Thank you.
Thank you, sir. Now I would like to request Dr. Samir Naik sir to deliver his talk. Over to you, sir. Dr. Samir Naik sir.
Hello everyone, and once again thank you this college that they've invited me for the second occasion. In the recent past, the first interaction that happened in December. Now this is for another topic, and thank you Dr. Ghorpade for a warm welcome, and thank you for providing me an opportunity to interact with the students and academicians and representatives probably from industry. So of course I would be giving a broad aspect of biotech product development in my presentation. So if you could allow me to give the right to share my screen so that I can start my presentation.
Sir, you are a co-host. You can share your screen.
Just a second, so I just opened my presentation. Just a second. Are you able to see my slides?
No, sir.
Just
It is not visible to us.
Yes, sir, it is visible.
Yes, sir.
okay.
Yes,
So
sir.
this is the slide. Okay, and I'll now switch to the PowerPoint mode. Okay. I don't know why it is not working, but I'll just move like the slides like this only. There's some issue with this. Okay, so like I made it a very generalized, broad topic is biotech product development from the quality and the regulatory perspective. And thank you once again for inviting me. I'm Dr. Samir Naik, Assistant General Manager from Serum Institute. So the outline of my talk, I divided into 4 different topics. That is just a brief introduction about what are the different biotech products, their deliverables, overall product development stages. Then a brief discussion that we would talk about the product development cycle, process development, costing, feasibility, and R&D. Then the third part, which is from my side as a quality person. So you just give what is the concept of quality in process testing, stability studies, validations. And the last part is the regulatory aspect. What are the different regulatory guidelines, the laws prevalent in India as well as at international scenario. So I will just briefly discuss because each on this topic itself is uh, a very big chapters, and then we can have later on if you know uh, we get a chance, then we can definitely have more discussions on each those topics in depth, maybe in subsequent series. But today I will just restrict myself to this particular aspects. So, what are the what are the products that are covered under biotechnology and biotech products? So here, the health and biotech products that specifically I would like to mention. So in case of health product, as everyone know, the pharmaceutical products are there, medical devices, diagnostics, in vitro diagnostic, biologics and biotechnology, nutritional product, cosmetics, and veterinary products. So these are all the different health products are there. Within biological products, which can be divided now into different products, like everyone knows about the vaccines nowadays because of the pandemic. Then biotechnological products such as enzymes, then the health product which are derived from microorganisms or using by cell culture methods like biosimilars, monoclonal antibodies, etc. Then blood derivatives. So blood derivatives include like coagulation factor, factor 6, factor X, which are important for the blood coagulating agents, or you can have some other products derived from blood. The whole blood, again, it is coming as the biological product, which either in the form of plasma, serum, or whole blood is given. Again, there are some rules and regulations which everyone probably are aware in terms of the blood banks. Blood components like plasma, serum, albumin, platelets. So these are all different blood components. The upcoming field for the biological products is like somatic cells and gene therapy, particularly for the stem cells and stem cell therapy. Then xenotransplantation, that is the transplantation of certain organs, tissues, or certain cells derived from other organisms. Allergic extracts, which can be having some medicinal properties and values, and tissues. So these are all different products are covered under a biological or biotechnological aspects. So what makes a product, what are the drivers? So for the product to come into a market. So one of the important aspect of the product driver is the marketability. That any product that is developed, whether it is a vaccine, pharmaceutical drug, or any of those categories that what we have seen in earlier slide, it should have some market. And now, as you know, the vaccine has a tremendous market in recent days because of the pandemic. And of course, it's a medical need at the point of time. Then it is a recommendations from opinion leaders, the technical feasibility of the product, the regulatory environment present in particular geographical region, and the public opinion. So these are all different drivers for the product which would come into the market. And of course, like I would not go into the details of each and every aspect, but just will tell that these are the related point which makes the product successful and the background for the product development. Okay, so these are the product development stages, whether it is a vaccine or any other biotechnological product of health importance. These are classified as the discovery. From discovery, then it goes to a pilot plant. So the scientists or the R&D industries or R&D institutions are involved in different types of research, either basic or applied research. And those who are working for the health, like biomedical research or health related topics, they found certain molecules or antigens or the lead substances. So the discovery is coming from those R&D laboratories. If those R&D laboratories have demonstrated successfully the different characteristic of a product, then that can be taken up for the pilot scale. From pilot scale, then it will go to an early clinical stages. So these are all early clinical stages, like include preclinical stages such as toxicological stages or maybe early phase 1 studies to see the safety of a product and to certain extent the immunogenicity. Then it will go for a scale up, and then you have a late clinical stages like the phase 3 clinical trial, where can say the efficacy is done, and then the product is successfully marketed. So since I'm from the quality division, so I have included the three important aspects for the analytics. The one is the animal model development just after delivery. So this animal model would give the correlates of immunological reactions or the pharmacokinetics or pharmacodynamic kind of a models, which would give certain lead how the molecule is going to behave in animals if it is given by the specified route. Then during early clinical development, you develop the different analytical methods required for the analysis of the drug product. And during the scale up, you have to validate all the assays for the analytics and the release of the drug product into the clinical as well as in the market. Of course, when the product enters into market, it does not necessarily mean that the clinical trials are stopped. There is one more clinical trial that happens after marketing when the market is there. That is called as a phase 4 trial, which is called as a post-marketing surveillance, which is either done by the organizations or the contract organization. And in most of the cases, this is done by independent bodies and the regulatory agencies such as WHO, UNICEF, or here in India like ICMR. So these are all different bodies that are involved to see how the product, when it enters into market, how it is behaving, whether the people are being protected, and the desired result that what observed in the clinical trial is also observed when the product is coming into the market and given to all individuals as a common individuals. So, when we want to develop a product, it involves, it's a multidisciplinary stream, and there are many people who are involved. Basically, for a product, the core team is involved as a project leader, project manager, and the regulatory representative. Then you also require the different sub teams, like say for example the CMC QA QC teams. Then you require marketing teams. You also require the uh, pharmacovigilance clinical team. You also require these different sub teams such as the assay development, preclinical biology, research and discovery. So basically, if I just want to briefly tell that my job role is uh, the lower hemisphere of this circle. So where I am involved in all this activity as a sub team is CMC sub team, as well as the assay development, preclinical biology, research discovery, PKPD studies, and some regulatory aspects related to the development, the R&D, and the support. So, of course, like if you see, it's all these areas are interdisciplinary. It's not that the product development is happening only because of one individual. It is the effort of all the individuals from different expertise. And then when you say that, what is the role of a marketing person when we are developing the product? Of course, a marketing person is the major driver for a development of a product, which are giving very vital inputs about what should be the dimension of a product, whether it should be in the glass vial, whether it should be in the ampules, whether it should be in the bottles, or it may be in suitable container, what should be the delivery of the devices that needs to be provided, or how the product needs to be delivered, what is the market feedback, whether we develop a product in one particular fashion, then whether the common people they would accept or reject, what is the regulation says, whether it should be packed as an individual container or as a multi-dose container or single dose container. So all these factors, all these different aspects are coming as a feedback from market analysis and the marketing team. Okay, so of course they are important for product development also. Now let us go into one step by step and dissected the product development cycle, how the product is developing. I'm sorry that the spelling of concept and the product, the last part has been because of some reasons they're not being visible. My sincere apologies for that. But basic is like from a concept, then it goes to a proof of concept, and then it goes to a product. So that's the basic development cycle. So how it goes? So you have the researcher who develops the molecule, as I said, in different R&D institution, R&D laboratories, or even in industry, whatever the research is ongoing. So after the successful research, then you develop it. It is also important to perform some kind of preclinical studies and early clinical studies at research level. At the simultaneously, it will also go for development, clinical, and regulatory affairs. So these are all different stages. Like you have the preclinical, phase 1, phase 2, phase 3, then you have a dossier and the launch. And then you require the efforts of all these people from research, development, clinical, regulatory affairs, marketing, production, strategic marketing, business unit. At different stages, they will come into picture. So if you could see the strategic marketing, they would come right from the beginning to the launch of a product. The R&D people will be involved up to the clinical stages. The process development people will be involved up to the phase 3. The clinical trial people will be getting involved from phase 1 to phase 3 up to the dossier launch. The regulatory affair individuals would also get involved themselves from phase 1 or preclinical studies till the product launch, and then their job also continues even if it is in the market. So this is how different teams that are operational for the product that is under development and the product that has been successfully launched. let me tell you that although it appears to be very simple, that it is going from a concept to a proof of concept, from a proof of concept to a product, although these are only just couple of steps, but if we see the product that has been developed recently, or if you see the history, if you take an example of COVID vaccine, we had approximately 70 to 200 research papers which were claiming that this is the putative or the successful candidate uh, in the research, but only 70 candidate as made as a conceptual vaccine candidate. Out of 70 candidate, some of them, went up to only experimental stages and the preclinical stages. Some of them went up to the proof of concept. And out of 70, at this point of time, there are only 5 to 6 products are in the market which have successfully completed all these stages of development after a regulatory aspects and consideration by the government of the respective countries as well as the international regulations, and then the product in the market. So probably those who have attended my previous lecture where I said that, of course, this is an unprecedented time, and during this unprecedented time, the vaccine development, if we see out of 70 candidate, it's only 5 to 6 products are in the market. So that does not mean that rest 60 to 65 candidates were not good. They were good, but due to some or other, stages of development, the technologies, or some kind of the hurdles that they faced during this particular development, these 60 products, they did not come to the final marketable product. Of course, few people, they backed out. Some of the people, they are improving their strategies, and again they are coming into these particular cycles so that the product is coming as a marketable product. Again, like if you see the circuit diagram here in the product development and the roles of product development and how many individuals that are involved and their connections. So this is kind of a circuit diagram that I want to give it to you. This is a part where the molecule has been discovered, and which is a part we are into a preclinical and a developmental stages. So in this particular development stages, if you could see that the core of this development is the GMP manufacture and the quality control departments. So the process development and the quality control are playing the key role, whether it is coming from a discovery and from the process manufacturing. So the supports are coming like from analytical techniques, the evaluation of formulations, evaluation of cell lines, product stability data. So all these aspects has been regulated and tested and released by the quality control department. You have the clinical team who are working on the toxicological studies, preclinical safety studies, device of clinical protocol, development of clinical assays, and other things. And all these things are monitored by two important bodies. One is the quality assurance and the safety data monitoring board. All this activity during this development is then submitted to regulatory individuals for regulatory review. Based upon the successful review and the data generated because of the efforts of all these people, the regulatory review will permit for the clinical trial. So it's not that from discovery it is directly jumping into a clinical trial. The product has to undergo all these cycles of development, the interaction between the teams, departments, and different subject matter experts in that particular field would, when they significantly contribute for a product, then you get a successful regulatory review, and after that regulatory review, you are allowed to do clinical trial. And of course, we don't know what would happen in clinical trial, whether that particular drug or a vaccine or a product, whether it will be successful or unsuccessful. Okay, and all these are things is driven by two important aspects: is the intellectual property generated through the development and its freedom to operate so that it will go into the market, and the medical need and the commercial opportunity. So these are the two important drivers that the product will very quickly develop and then enter into clinical trial. So these are the two important aspects. Again, coming into the part of manufacturing process development. So, again, there's an interaction like what is the process R&D, analytical R&D, GMP manufacturing, the scientific activities and costing part. What is important to understand during the development and the people who are involved in development of a vaccine, what is important is the cost. Okay. Now, when we develop a particular vaccine or a product, the very important and the first part is that what should be the cost. So the cost would depend on the raw material, the type of cell lines or the substrate that we use, the antigen, the type of product process that we develop, the amount of individuals that are involved, automations, technologies that are involved. So these are all different factors which are governing the cost. And if you could see that all these aspects of drug substance or drug product manufacturing is governed by two important factors, is the time and the cost. So if you take a lot of time to develop, the increasing the cost. If you are very quickly developing a particular product, you would save a lot of time, as well as you can get some kind of a cost benefit with respect to a product. Okay, so let me just see. Okay, so from this, I would shift myself from the development to the quality. So when we say that what is quality, the concept of quality, of course, this part we would discuss very briefly. So the quality is an ongoing process of building and sustaining relationship by assessing, anticipating, and fulfilling the stated and implied needs. According to ISO 9000, the definition of quality is given as the degree to which a set inherent characteristic fulfills the requirement. The standards also define the requirement as the need or the expectations from the law and the regulatory compliances. The American Society of Quality is also, the definition is given. It's a subjective term, wherein the characteristic of a product or the services that bear on its ability to satisfy the stated or implied needs, and B part is the product or services free from deficiencies. Is again a general definition of a quality. So as you know that each and every product, when it is released in the market, it has certain kinds of specifications. Like, if you purchase, say for example, mobile phone from the electronic shop, you are having certain predetermined specification, whether this particular, the very first nowadays, the specification that comes in your mind is whether your phone is a normal phone or it's a smartphone. So this is the first specification that you set when you purchase. The second specification would come whether it is a touch screen, whether it's a Gorilla Glass, whether it has got an Android system or whether it is an Apple system. So these are all the specifications that you are setting up. to yourself and based upon your specification, then you are purchasing. Similarly, when you go to the shop of a mobile phone, then you see the technical review and then you see that different specifications are mentioned. Similarly, when you purchase one particular drug, whether it's a tablet, a simple paracetamol tablet, or the vaccine, there are certain things are written on the label. So this label also states what is the composition of the drug or the vaccine, what are the ingredients of the vaccine, where it has been manufactured, what is the quantity of those ingredients, and then there are other parameters like what is the license number, where it has been licensed, what is the batch number, what is the manufacturing date and expiry date. You see, these are all the results. So basically, when we go for a label of a product, it gives that this particular vial or this particular tablet contain XYZ amount of the product. So that is the specification. So when you test that particular product, it should not contain the value or the amount of that particular active ingredient. It is expected that it should not be less than what has been claimed on the label. So the specifications are defined as the list of tests, references, analytical procedures, and appropriate acceptance criteria, which are either numerical limits, ranges, or criteria for that particular test. And it also established the criteria to which a new drug substance or a new drug product should conform to be considered as acceptable for its intended use. So what is important for a specification or for drugs or the vaccines is conformance to the specification. So when we say that this particular vaccine is sterile, so that's the specification. So the vaccine vial should conform to its specification of sterility test. Similarly, like there are certain set of tests which are applicable for the product. So that particular product should comply the stated predetermined specifications. So in short, specifications are the critical quality standards that are proposed and justified by the manufacturer and approved by the regulatory authorities as the conditions of approval. And of course, one has to make a full justification of each and every selected specification for that particular product. Mind well, all the product specifications are product specific. However, there are certain general specifications which are listed in general expectations by regulatory that I would come in my subsequent slides. Any questions so far? So I hope that you're able to get this information. So before we go into the manufacturing process, do you have any questions or I move ahead?
So we can move ahead. We will
Okay.
take the discussion panel later.
No issues. Thank you. Okay. So basically in vaccine manufacturing, there are two important aspects. Even for the drug development, there are two important stages of manufacturing. One is called as the drug substance manufacturing, and the second step is the drug product manufacturing. So in case of drug substance manufacturing, then you have a master stock of one particular antigen, from which you prepare a working stock. You scale up that antigen to at an industrial scale so that you get a production culture. This production culture is, I took an example of the master stock here is the cells that are used for preparation of vaccines. So just for an example, the Viro cells are being used for vaccine manufacturing. So you have the master stock of Viro cell, which is brought up to a production culture. Then you have a virus. Its working stock is used for the infection to the production culture. You incubate this production culture to get an harvest. And then you have certain quality control tests that are being described here. Coming to the drug product manufacturing, so from the previous slide, the harvest is pooled, to call as a pooled harvest. Then you have optional as in downstreaming process. If it is required, you carry out some downstreaming process. And these downstreaming process are typically either centrifugation or chromatographic techniques such as ion exchange chromatography, size exclusion chromatography, or simple filtration or concentration by diafiltration, or it may be the reverse osmosis. So these are all different examples which are described in details in the literature. So use all those permutations and combinations of the techniques so that you get a very good design of a downstream process, so that you would get and able to attain a product in such a way that it will retain its maximum potency and the minimum level of impurities. So the design process should lead to a good yield with minimum losses. And of course, the process should be such that it should remove and reduce the process related impurities. Then after this downstreaming process, you prepare the blend. The blend is then filled into a vessels or the vials, and then it comes as a finished product. And at different stages of manufacturing, then you have a quality control departments who are playing their role to test and the release of in-process material, the final blend material, and the finished good material. And during this process, at each stage of quality control, whether it is at harvest or maybe at final product, you have the release specifications which are predetermined with predetermined acceptance criteria. And then one has to study the stability of a product as the intermediate stability, that is the drug substance stability, or the finished product stability by the prescribed norm. So that based upon these stability studies, one can determine what is the shelf life of a product. Using that shelf life of a product determination, one can assign the expiry date or the shelf life to the product. So what are the different quality attributes for a product? So these are all generalized tests common for almost all vaccines. The product should conform the following criteria: is identity, the quantity, so what is the specific activity of the active material, purity of the product, and the quantitation of the contaminants. Then we need to characterize the active ingredient with respect to size, structure, or the sequences. Then the activity of the antigen. Now here in this case is either based upon the animal-based assay where we can have the immunological response. Based upon the immunological response and its assessment either in vitro or in vivo conditions, we would come to know what is the activity of the molecule that is eliciting an effective immune response or not. The potency is important for vaccines. The vaccine should have the minimum required potency where it has been demonstrated the effective immune response and the protection against a particular antigen or the pathogen. And other important microbiological aspect, particularly for vaccines and sterile preparations, is microbial sterility with respect to bacterial and fungal contamination, pyrogenicity, toxicity, and general safety. So these are all the important attributes which are the quality parameters for in general for vaccines as well as other biotechnological product of health importance. Of course, I would skip these slides of method validations because I think this would be taking a lot of time. Just to give you a brief idea that during the development stage and the late scale-up stages, we have to validate the analytical methods. Of course, the expectations from different agencies are different. For some of the product, it is expected that the method has to be validated before the start of clinical trial. Some of the expectations are during clinical trial, or some expectations like they are during or earlier than the phase 3 clinical trial. So for any method that needs to be validated, like say for example, it may be sterility, potency, or any immunological reaction, the method has to be validated with respect to all these parameters, and only the validated methods are allowed for the release testing of the active molecule. I'll again briefly go through this quality management system principle, a part of quality assurance. So in the quality management systems, you have three different sections where we have classified as an incoming material, the process material, and the material which is going as the finished good material. So the incoming material, like the raw materials that is required for the manufacturing, so it should have the sampling and initial process check. Again, the quality control department or the quality systems is important for the release of this incoming material. Then this incoming material, when it is checked and released, it will come into the process material as a part of a routine production process. And then based upon that particular process, the functionality testing and the inspections would carried out at different stages of manufacturing. After its successful release, the material will come as the finished good. This finished good would again be tested, monitored rigorously with respect to quality parameters, and then only it is released into the market. Just to make it slightly simpler, you have the procurement where it is coming from the supplier. So the different aspects coming from the supplier quality is with respect to the supplier audit, contaminant and failure analysis from the raw material supplier, then supplier corrective actions. So these are all expectations from the suppliers and the procurement agencies. Then the manufacturing, where we have an internal quality with respect to different other quality parameters such as internal audits, inspections, the change control and the deviation systems, the corrective and preventive actions, quality control charts and quality scoreboards, safety management. So these are all different parameters being monitored rigorously by the internal quality assurance team. Then it goes for a distribution in the customers, and again the quality comes into a picture with respect to the customer quality. And this is very important for any molecule where we face, or whether you get any kind of any customer issues, complaints, any failure, any kind of an issues that are related to the product. Say for example, we get a lot of queries when the product is coming into market, and of course some of the queries are very important, serious and cognizable. some of the queries that we receive from people are like the very craziest queries that we get. And especially under this pandemic, people are learning so many things. And I know that just to make it on the lighter side, that a new university has come up from last 1 year, and there are several graduates, and some of them are doctorate from this university. We call this university as WhatsApp University. So people get enormous knowledge from this WhatsApp. They read something and they ask a lot of questions which are relevant and which are very irrelevant questions. So, uh, like uh, most of the queries coming, like if I can give an example, of course, I would make myself very sober. Of course, we got some craziest queries that in some of the queries, I would give later on some example. The more frequent queries coming from the male strain of Homo sapien is that after vaccination, can I drink alcohol? I cannot drink alcohol. So that was the major type of queries that we get after the product launch. And of course, some of the queries are, such that they should be published in one of the column that is famous in Pune Mirror. I will not say, but Dr. Vats is the person who answers such queries. So we get even those kinds of queries after vaccinations. Okay, coming back to this particular part, that the consumer complaints and the issues are important, and they are the actual reflection of a product, how the product is behaving, and what are the feedback that we are getting, whether the product is really effective or does it have some issues. Again, there are certain norms, procedures being prescribed as a part of a customer quality program. Okay, now I'll just move again to another aspect of the development is regulatory consideration. For vaccines, these are all important regulatory considerations. I just give some of the examples like the World Health Organization, Food and Drug Administration, PAHO, that is Pan American Health Organization. Then you have Health Canada, EMEA, MHRA, ICH. So these are all different regulatory agencies and regulatory guideline providers, that what should be the quality, what should be the testing program, the manufacturing quality, the manufacturing norms, and all those things are being prescribed by all these different agencies and the expectations from the regulatory agencies. So, what are the different guidelines that are available either in local or in the global scenario? So here is like the different guidelines in the form of pharmacopoeias, where for drug or some chemical molecule, it has come as the general or specific monograph for that particular product described in Indian pharmacopoeia, either in European or British pharmacopoeia, US pharmacopoeia, Japanese pharmacopoeia, Health Canada guidelines, and you have the local or national FDA guidelines. And the local authorities here in India, so the regulatory agencies are the Drug Controller Governor of India, the Ministry of Health, and the National Control Laboratories, and the National Institute of Biology. So these are all the two important agencies who are releasing and testing the quality of a product before it comes into the market. So in case of regulatory affairs in India, I will just briefly come and tell you, all these are governed by the drug, and the vaccine is governed by important act, what is called as Drug and Cosmetic Act, and it has got different schedules like Schedule M, Y, H, P. So depending upon the, like say for example, Schedule M is for the manufacturing, Schedule Y is for the clinical trials, and similarly for others, they have been prescribed. For even the clinicians, clinical health practitioners, etc. So every one of us is bound by the law of the land, which is a Drug and Cosmetic Act, and the regulatory requirements as given by the CDSU, by the Drug Control Governor of India, the local Food and Drug Administrations, FDA in Maharashtra is FDA Maharashtra. So these are all the guidelines that we follow, and of course, like there are certain GMP norms which are accepted globally. So even those are applicable for the vaccines. And needless to state that. I will skip these two slides also because I think I completed my time of 1 hour. Because this slide will again take another 10 to 5 minutes where I have to describe the Schedule Y, the requirement of clinical trials according to the Drug and Cosmetic Acts. Basically, for any clinical trial, what we need to submit is the manufacturing site and manufacturing site. We have to submit the CTD Module 3, and based upon the review and other thing, the clinical trial permission is granted as per the Schedule Y, and then the clinical trial has to be conducted as per Schedule Y. And then after successful completion of a clinical trial, we have to go to the next clinical trial, or we have to market the product. Of course, there are many intricacies are there with respect to the laws and regulations in the clinical trial and clinical trial applications. But I would restrict myself to this particular slide, that just to make you aware that clinical trials in India is governed by Schedule Y, and any drug that needs to come into the market, whether it is a drug or vaccine, it has to go through at least some clinical trial, and it should prove at least the safety and immunogenicity in case of vaccine, and then only that particular product is allowed to get into the market. Well, thank you so much for your time and patience. If you have any questions, I'll prefer the questions.
Thank you so much, sir, for enhancing our understanding pertaining to the process of drug and vaccine development. Thank you so much. Now, I would take the opportunity to introduce the next eminent panelist, Dr. Arun Kharath, sir. Dr. Arun Kharath, sir, is Distinguished Professor in Microbiology. He has more than 20 years of teaching experience in various institutes. Presently, he is working as a professor in Microbiology, School of Life Sciences, JNU, New Delhi. He did his PhD from IIC Bangalore in year of 2000. He was awarded by postdoctoral fellowship by French Ministry of Science for 1 year at France. He was also awarded by Rockefeller Fellowship for 7 years to do postdoctoral work in Rockefeller University, New York. He has 35 international publications. Under his guidance, 8 students have completed their PhD. 3 of them are foreigner, 1 each from Portugal, Germany, and Yemen. Currently, 8 PhD students are conducting their research under his supervision. He also delivered national and international talks. So with this brief introduction, I would like to request Dr. Arun Kharath, sir, to deliver his speech. After his lecture, we will take the discussion. So over to you, sir, Dr. Arun Kharath, sir.
Thank you very much, the organizers. But the biggest task that I have, and I'm afraid and scared also, that largely the topic is going to be redundant and very much like going to be repetitive to large number of students, because I'm hearing Dr. Naik's presentation. Everything is covered, so it just would be a different slide, but it will not add anything, literally anything to the presentation. So because of the overlap in the topics given to me, this is going to be the case. So I'm sorry for that. Let me share the slide. Are you able to see the slides?
Yes, we can see your slides, sir.
Okay, okay, let's see. Yeah, so I'm going to take FDA, that's US FDA as a model, and going to give very fundamental outline, not in a great detail as Naik has given so many details to you. But I thought since it is a UG PG student background, I personally wanted to make it very introductory and very simple so that the interest among the student will be generated to mobilize themselves into the drug development or drug designing the process. So I'm going to take a review of the process, like what are the objectives first of all. So it's a process, one has to learn the process which is involved in the drug discovery and the drug development. So that is the objective of our presentation out today. And what are the phases are involved? I will be able to define to you the different phases which are involved in according the Food and Drug Administrator approval to any of the drugs. And explain the role, different roles which are given to the Food and Drug Administration. Normally, why I took the FDA, not DCGI, is because DCGI is only for India, while FDA is for the global, and whatever is approved by the FDA in general is considered not just for the United States, but also it works for the global review. And this is more or less a comparable review process with DCGI also. So basically why we talk like, especially in the last April, everybody was thinking why there is no another drug, why there is no drug for the virus, and still talking. We are still talking like why there is no drug for COVID, or why there is no vaccine, why there is no this, why there is no that. But first of all, drug development is not that easy process, and it is not somebody would dream and it would happen in one night or 1 week or 1 month or even 1 year. Although we have a historical moment that we are all vaccinating, and some have vaccinated, some will be vaccinated, that almost more than 10%, 12%, 18% of Indians have received at least one shot of vaccine. That is world's history, world's historic vaccine that was developed only in 11 months, which generally is not a common case. So for developing a new drug, it is a complex process because it includes several different component stages, which Naik has already covered in his presentation, and it is a costly process, both on time as well as money. It probably would take anywhere around a decade and maybe over $350 million in order to identify and market a drug from the laboratory to the pharmacy. So it's like what we call like lab to land in the agriculture, and your lab to the pharmacy shelf, that is over the counter or up to the prescription in the pharmacy. So it is a process, it's a journey that takes a lot of time, as you would have to understand. So it's basically a patience demanding process. One needs to have a huge amount of patience. One needs to have a lot of logistics, and then most importantly, a targeted study. So research and development basically includes the discovery of a drug, and there are mainly two main areas, which is a preclinical studies, that is a laboratory research, animal research, and then basically developing the same material molecule or that drug as a marketable. So initial research, fundamental research that goes into defining a high throughput in these days, particularly because it's not merely like synthesizing 10 or 100 components. I recently read an article in Nature. They had flipped through almost 9.6 million molecules in order to search for one new antibiotic. That 9.6 molecule, let me summarize, it would be 96 lakhs. So if you add 4 lakh, it would be 1 crore compounds they have screened. So you can imagine how much strenuous, how much intense, how much time consuming the process, even at the preclinical studies, it can be. And once you come up with a molecule, then the beginning of the process is, if that molecule is a druggable, then you have to enter into the second phase, what is called the clinical studies, where basically the safety, efficacy, side effects, everything is going to be studied beyond the animal. So only a few thousand compounds that began in the laboratory testing will make it to a human testing. So I'm putting it a modestly few thousand, but I just gave you an example of one of the new antibiotic that is published in Nature in December 2020, that they targeted isoprenide synthesis in escape pathogen Vibrio, Mycobacterium tuberculosis, and the malarial parasite that is a Plasmodium falciparum, wherein 9.6 million. So you can see like a thousands is nothing in front of 96 lakhs. So what is this? Like you have a large number of compounds to flip through. Then you come up to the drug discovery search, that would be like docking. Nowadays, you have a computer-based modeling studies are saving a time and allowing you to search a large number of libraries. Either they are synthetic libraries actually, or you may have chemically in silico designed molecules, and you can predict structurally, and their molecular docking will allow you to envisage the activity, efficacy of the compound. So it could have 5,000 to 20,000 compounds, which comes after basic research into the drug, which will enter into the preclinical research almost a few hundred, and in clinical research goes into a single digit. And finally, it is a possible that one molecule out of those thousands, tens of thousands may get approved. It's not always that the one will get approved, especially in the field of antibiotics. It has been seen since from the penicillin discovery till today that an antibiotic that is brought into the market has been found to have a resistance by the microorganisms within a year, or sometimes less than a year. So after so many years, after so many people, different divisions, so much of money, so much of energy, so much of time has been spent. And what happens is that the moment you marketed it, it has failed the whole purpose of it. So this happens, and that means a new cycle restarts of a new search, and again a search for the molecule. And not just an antimicrobial agent, but the anti-cancer agents are also same. You will find like a lot of anti-malarial parasites, similar search history has been repetitively happening. So what happens here? Discovery of and origination of a new compounds that become a drug candidate, which is normally called a laboratory research or a basic research, which may take a PhD. And you know that sometimes not just a single PhD can give you a drug, especially. And this new compound efficacy and safety has to be then studied another PhD period. So you can have like already you have a 7 to 8 years spent in order to find out a molecule has a safety or a molecule can have efficacy. Then it is going to enter into the clinical studies, which could take a couple of years. And then again, that would be done into the humans particularly. And then it would be applied for the new drug applications. The review process begins, and the review process will complete around 1 more year for that administrative regulatory process. And then finally, the approval and launching would be taking a step. So in the case of clinically, you have phase 1, phase 2, phase 3. Basically, these are the main phases. And as rightly said by Naik also, phase 4 is yet another thing. Now again, repeating the same. Basically, you start with the basic research, then early discovery, then it is into the preclinical, comes into the indigenous new drug application. And once you are IND application, then it would enter into the preclinical to the clinical development, wherein you have a phase, different phases. Again, after that, a new drug is submitted for the approval and BLA application. That is then the FDA will take a review and it will enter into the next phase, and that will be the monitoring and launching. Then that's a surveillance. Now, what is the Food and Drug Administration role? Basically, it is required to review and approve a set of new drugs in the United States and probably the global also. The FDA reviews and evaluates new drugs which are based on the evidence. It has to be an evidence-based, not just a computer evidences are sufficient. You have to have substantial evidences in the cellular level, in vitro level, then into the animal level, then preclinical to the clinical level, and then in human. And afterwards, it would be establishing itself as a molecule. And especially during the clinical phase, the FDA will closely monitor the development and the results, efficacy, and the safety of the molecule. So this slide is again a repetition that was shown by Nair. So you have in the preclinical research, you have basically synthesis and a purification of compound. I'll show you in a couple of next slides. And then also you have animal testing, can be of two types. You can have short term impact being studied, or a long-term impact can also be studied. Sometimes you will see the efficacy directly relates to the clearing of the symptoms or the conditions that is reversed with the use of animal, with the use of the drug molecule. That's a short term. But a long term, like whether in the whole generation and the subsequent generation, animal continues to respond to the same way. There are no mutation, there are no genomic instability, there are no any side effects. Those are the things like in the human. Animal also, the long range models are there. The matter has to be approved by the Institutional Ethical Review Board. That is, in India, it's not IRB. In India, it is IERB, Institutional and Ethical Review Board. Particularly in the clinical research, it cannot be without Ethical Review Board. In case of animal, you have a IAAC, that is Institutional Animal Ethics Committee. So it's a more of ethics, whether it is a preclinical or into the clinical, especially in the case of India. Now here, in the case of clinical phases also, you have 3 phases. Phase 1 is on the healthy subset, and this is a very limited number, sometimes in a single digit. Phase 2 could be the patients who are being treated. Some set of patients are being treated with the drug, and some set of people are also treated, patients are also treated with the placebo. So placebo is basically looking at the non-drug molecule to ensure that the changes or whatever effect that we see are indeed due to the drug molecule, and there is no yet another some sort of psychological or psychophysiological effects. And then it enters into the next phase, accelerated development and a continuously back-to-back review process. Then it comes to the treatment of the new drug molecule, and parallelly a track is also being seen into the phase 4 particularly. So why drug? When you want to define a drug or develop a drug, it becomes very important that one should choose a disease. That disease could be a genetic disease. That disease could be an endocrinological disorder. That disease could be a lifestyle disease. That disease could be a disease due to the metabolic troubles such as insulin, or that disease could be an infectious agent that could be bacterial, fungal, parasitic, or COVID like a viral disease. Or you have to first identify a disease that you want to find a drug against. So pharmaceutical companies are the commercial enterprises in choosing and endeavoring the same. So pharmaceutical companies will therefore tend to avoid products with a small market. For example, disease which only affects a small subset of population. It is needless to say that today you have dozens of companies involved in making, global I'm talking, in making the different vaccines against the COVID-19, because it's a global population, it's a pandemic. So if it was only a small part of the world that was going to be affected, only the few companies in those countries would be showing the interest. So Pharmaceutical companies will also avoid the products that would be consumed by individuals of a lower economic status. Usually, that is also it's basically the sometimes it comes as a enterprise a matter, a decision which only affects to the third world countries. I don't have a shame in saying that third world countries have always been serving themselves as the clinical subjects without qualifying, without knowing they are the clinical subjects. Very recently, in last or in the 21st century, especially after 2007, the clinical research has become more popular and announced noticeably in India. Otherwise, before 2000 period, people were given the drugs in India while they were being checked out. While on the other hand, the developed countries would not even prescribe, would not even touch upon. Even today, for example, an antibiotic is not given in the developed country to an infant unless he or she is 9 kilo weight or 2 years age. However, in India, everybody knows that immediately after birth, within a few hours, administration of antibiotic is not uncommon. However, whether it is indicated, whether it is not indicated, and COVID can be an added flavor. Now you see, the plasma therapy became so popular, and in this week, plasma therapy has been proven to be a failure. Remdesivir came. India was the first country to approve Remdesivir for the use. And now very recently, people have realized hardly Remdesivir makes any difference to it. Then we go on naming it. Steroids were become so popular. Everybody, including half of the people sitting out here, must have seen videos of different Western Indian originated doctors saying that 10 रुपए की दवा है, 10 रुपए का इंजेक्शन है, ये सिर्फ कर लो. अभी क्या हो गया, म्यूकोरोमाइकोसिस से महाराष्ट्र जूझ रहा है. And why mucormycosis? Oxygen makes it humid and dexamethasone makes it immunocompromised. A beautiful environment to grow the mucus. So here also, these patients have been used more or less to create or generate a data. And today, no Western country would combine dexamethasone or other prednisolone or any kind of a steroid along with the oxygen unless it is indicated. यहाँ पे लोग ऑटो में, कार में, घर में अपना ऑक्सीजन सिलेंडर लगा रहे. So this is a kind of a situation we are talking. So that is why the big companies, popular pharmas, won't be making it for the low economic status. Now, most research is carried out in those diseases which afflict the first world countries. That's basically the developed countries. Cancer, you would know that a large number of molecules have come up and large number of amount of years, over 50 years, cancer has been endeavored. Cardiovascular disease, depression, diabetes, flu, it has become a common. Migraine and obesity are a few diseases that we could talk about. But you would see there is no malaria. There is no malaria, which is a very endemic to the African countries and India-like or Indian subcontinent-like countries. So drug targets basically specific macromolecule or biological system where the drug will interact with. So the gene or the protein or the metabolic pathway or a particular pathway step we have to target, the drug will bind, drug will interact, and favorably or unfavorably process will be modified. Sometimes this can happen even through an incidental observations. For example, penicillin. Penicillin discovery was not a purpose discovery, it was an accidental. And large number of Nobel Prize are also an accidental discoveries. So here is an example. So you could see that a baby's face with like a pustules all over the areas. And now this is like a Vialogen kind of assay shown. So you have to have an assay developed in order to see the screening of the molecules, number of molecules for a particular step. So choose a disease, choose a drug target, and identify a bioassay. I have shown it as a 96 plate as an example, so that you would be able to see like 96 may say 94 may be the test molecules and 2 are the positive and negative control. So you could see the gradation of the optical density indicating whether it is with the negative control or the positive control YCIS. You would be able to see that. Find a lead compound out of those tens of thousands compound you started up with, and a structure that has some activity against the chosen target, but not yet good enough to be a drug itself. If not known the structure of that compound, first of all, find out the chemical structure, chemical nature of that lead compound. And if you already know the compound's structure, then try to modify it in a way so that the reaction can be, the speed of the reaction can be increased. So you can have a different kind of modification. Synthesize the analog of the lead. That's what I said it already. Identify the structure activity relations. Synthesize the analog of the lead. Identify the structural relations with the analogs also, or the homologs also. Then try to identify the pharmacophore. Particularly, what is a pharmacophore? The structural feature that directly is responsible for conferring the activity. Optimize the structure so that it will improve the molecular interaction, and thereby it will have a high efficacy of that particular molecule. Then it comes to demonstrating the toxicity. Initially, before animal, you could do that into the cell line, and after doing the cell line in vitro studies, in vivo assays can be performed using the animal model. So mouse, hamsters, guinea pigs are the various animal models, all the way up to the primates like monkeys. Now, if you first, in the preclinical or the this era, you have been able to find out a potential druggable molecule. So the first and foremost is to patent the drug compound and continue the study for the drug metabolism, and further go ahead with the testing of that drug molecule. So design a pharmaceutical manufacturing process, carry out the clinical trials, and after the clinical trials, which are successfully done, then the marketability to that drug can be obtained through the FDA or the DCGI in the case of India. So what are the preclinical trials? Synthesis and purification of the new drug. Pharmacology of the new drug, basically it includes pharmacokinetics. Its absorption into, correctly absorption into the targeted site and not all over the body, because then it would be another problem. The distribution of that molecule should be very targeted rather than going into the different areas. Otherwise, it will be a diffusion, and it would be another chaos in the body, and there will be a lot of noise over the signal. Its metabolism, whether it is a biliary route, renal route, etc., one has to find it out. So the kidney, the liver, and the gallbladder are the primary routes where this would be metabolized through. Excretion, it's a half-life. So that would, the half-life would decide the number of doses and the body mass index related issues. So pharmacodynamics, mechanism of actions, and it would estimate the therapeutic effect also. Toxicology will be conducted, including the carcinogenicity test, then the mutagenicity test, Ami's test, and the teratogenicity test, so that during the the gestation, there is no imparting any kind of anomalies into the developing fetus or the embryo. Efficacy studies can be carried out, or generally are carried out, onto the animals. These are all the preclinical trial, and that is called IND, that is investigational new drug, which is meant for the application for the permission of administration as a new drug to be used onto the human. Outlines the proposal to use the new drug for human testing in clinical trials. Studies in humans can only begin after this investigational new drug is reviewed by the FDA and approved by the FDA. But prior to FDA, it has to be reviewed and approved by the Institutional Review Board in case of global perspective, and in the case of India, it should be approved by the Institutional Ethical Review Board. Now, the parallel system in India is basically the Director of Controller, Controller General of India, that is a DCGI. In case of clinical studies, as Naik sir has already mentioned, there are like primarily three, but fourth phase is also there. So in the first phase, one would be looking more into the efficacy studies on the healthy volunteer that I already mentioned. And in the phase 2, clinical studies will be carried out on a slightly larger population, but still in a limited number of patients with a placebo effect also. And phase 3 comes as a comparative studies to be made onto the larger number of people, larger number of patients, and then new drug application can be made for the IND. Okay, IND then turns into the new drug application, and that would be subjected for the regulatory review. And in the phase 4, the further analysis continues. Comparative studies are again taken into consideration, and the registration and marketing is being carried out. So here are the 4 phases. Evaluate the safety, determine the safety of the dose, particularly identify the side effect, that is into a very limited, small number of completely healthy with the clinical studies criteria should be used. Almost 70% of the drugs would be moving to the next phase, wherein the test will be performed for its effectiveness. Out of all that, tools into the phase 2 will be moved into the phase 3. And this is where I would give you an example of the hydroxychloroquine was said to go to the phase 3, but which did not work very well, and it did not work as a COVID, one of the therapeutic. Now in the phase 3, again, it will reaffirm the effectiveness of the drug. In addition to that, the monitoring the side effects on a larger scale population, at least 1,000 or a few thousands will be done. And it would be compared with the other available treatment. Its efficacy will be compared with the previously or available other treatments, and information will be gathered, analyzed, and then interpreted accordingly. Out of all this, around 1/4 molecules from the phase 3 then are subjected for the phase 4, which will give an additional information after approval, and including the benefits as well as the best use of how the same molecule can be made as a best use. So, these are the same things. Timeline has been given to you. So this is what I was talking to you. In the phase when you have a very limited number, phase 2 you have a certain high number, phase 3 you have even more numbers, certain thousands of them, and in phase 4 you have generally a big population. But in between that, FDA takes a review. That means DCGI takes a review. As I would say now, very recently on the 7th of May, 2-deoxyglucose has already been approved as a COVID therapeutic molecule by DCGI, while it is under the phase 3 trial. So you can see, like, it is before the review is being taken, it has already been given. So it was made available from today or yesterday, if I'm not wrong. So phase 1, normally it is sometime in a single digits, not necessarily like 20 to 80. It is meant for the safety of the drug. Goal is to identify what are the major side effects, what are the major metabolism related, what is the metabolism of the drug, how this is being metabolized, and what are the excretory routes. So in order to figure it out, a gross anomalies if that impact, then that's what will be ruled out. So it would last some time, almost up to 1 year. About 70% of the molecules move into the next. Now, a couple of hundreds would be used into the phase 2 or participated into the phase 2. Of course, it's up to the consent and giving the proper signing their consent. Emphasis here is given along with the safety, also the effectiveness. So how effective this molecule is amongst the existing pre-existing molecule. So patients are receiving the drugs are compared to the similar patients who are also receiving the placebo or another drug. So you are going to compare it with the neutral treatment or with the comparable another molecule which is already being there. So you are going to check the effectiveness, efficacy of this molecule in the light of pre-existing therapeutic molecule. This can last up to 2 years. Almost 1/3 will pass this step. Now thousands of people will be recruited into the phase 3, and here again re-emphasis is given both onto the safety as well as the effectiveness. investigates through well-controlled studies, different populations and different dosages, as well as uses the new drug in combination with the other drugs. So this is what the T2DOG, again, I would say it is not been told to use a standalone bed along with the other therapeutics on the program. So it can last up to the 3 years, and almost 1/4 to 1/3 close to that molecules will be passing this phase. Then that is a pre-NDA period, that is just before that FDA and the drug sponsors will be meeting together, and then there will be new drug application will be submitted. The formal step of asking the FDA to consider approving a drug for marketing will be taken into consideration. FDA has almost 2 months to decide whether it will be filing the fit for the approval or not fit for the approval. If it is found that if filed, a review team is assigned to evaluate the new drug. The review team evaluate the research on the safety of the drug and its effectiveness. The FDA reviews the information to go on to the drug label. It inspects the facilities where the drug will be manufactured. So it is very important, the companies or the site where the manufacturing is done. So the prerequisites are properly made available, and also the storage, the capacity, and all other the microbial contamination or other chemical contamination have to be removed or made, ensured that it would be free from any kind of a contamination. Now, if the application will be classified as approvable or the not approvable, that is after visiting the site and everything would be then taken into final decision. If approved, the FDA will request additional information from the sponsor, and the NDA will again take a review once the sponsors give their information. Now, following the drug approval, sponsors of the drug will be required to continually assess the drug safety in the coming days, because it is a basically evolving stage, and some points will keep on emerging or cropping, and that has to be taken into consideration, not to be ignored. Now, the post-marketing surveillance of the drug, that is what is the phase 4, will be continually assessed for the safety of the drug molecule. So this is what we are talking now in the light of COVID. Lot of therapeutics have been said not to be effective, because this more or less now they have turned into the phase 4 kind of interpretation, and may include incidence and severity of rare adverse reactions also. Cost-effective analysis would also be done into the phase 4. Comparative trials, if there are like contemporary molecules available or the manufacturers available, and the quality of the life studies would also be taken into consideration in the post-marketing surveillance study. In the case of DCGI, has the same role in India while granting a drug usage within the India. So this is the last slide showing that drug discovery that starts in the laboratory and development. Initially, it is a preclinical research, then enters into the clinical research, then it would come to the review process, and then the individual drug becomes a new drug application, and a new drug application would be then applied to the DCGI in India or FDA in USA, which would be reviewed. Then the sponsors meet will be conducted, the manufacturer site will be visited, the additional information will be collected, it will be again analyzed, and all data and safety effectiveness measures are taken into consideration while granting the permission, and post-marketing monitoring for the further surveillance, and if any new point comes up, cost effectiveness and any side effects are being taken into consideration. I will stop here out, and if there are questions, then we can possibly answer to that. Thank you very much. Hello.
Hello. Yes, sir.
Yeah.
हाँ पंकज सर.
हाँ सर. So we'll take queries from the participants now.
So it is a very nice session by both panelists, and this session is open for panel discussion.
Yeah, Manisha Shinde ma'am has collected some questions. Shinde ma'am?
Yes, sir. Now
Yeah,
session
please.
is open for discussion. I would like to request all the participants to raise the questions or queries. You can put the questions in chat box or you can simply unmute yourself and ask the questions. तुम्ही इंग्लिश मराठी कोणत्याही लँग्वेज मध्ये क्वेश्चन्स विचारू शकता स्टुडंट्स तुमच्या सोयीनुसार. So
Hello.
yeah, yes.
Hello. Am I audible?
Yes, sir.
So I would like to question to Dr. Samir Naik, sir. Sir, for Covishield vaccine, as per earlier norm, the gap of between two doses was 28 days, but now it has now it is a norm is said that 12 to 16 weeks. So the individuals who took their second dose after 28 days, is it necessary to take second booster dose to them?
Sir, I think Samir sir is not online right now.
Is it okay if I answer that?
Yes,
Okay,
yes,
okay,
yes.
sir.
Most
Yes,
welcome.
sir.
Most
Pleasure, sir.
welcome.
Please.
Okay,
Most
okay.
welcome.
Most welcome.
See, I was telling you also the phase 4, if you remember my few of the last slides. Phase 4 is basically a continuation of the clinical research. And in the case of COVID, no matter whether we talk about Covaxin, Covishield, or any other vaccines, which are like over a dozen of vaccines are available globally. The biggest problem with these vaccines is that they have been made available within 11 months. So definitely they have not been gone through all the clinical studies, parameters, which otherwise are required. But at the same time, we do have to give it in contingency that the need of the hour was overwhelming. And if I say, like in the drug category, before answering to your question, Yadav sir, that A, B, C, D, the drugs are divided into 4 categories. A is not known to be unsafe. B is used in this condition. C is avoid for these people. And D is use only life is required. Now, COVID drugs or the COVID vaccine came under the D category because life was very important. I hope you are getting my point, right?
Yes.
So the vaccines were given
Yes.
permission right into the clinical phase 3. Now, Virapin, which is coming from the Zydus Cadila, is again in a phase 3. Your Covishield was again in a phase 3. And Covaxin was again not yet in a phase 3. So you can imagine, and India has not yet published the data about the efficacy and the safety, whether it is a Covishield or the Covaxin. So Covishield data was taken from UK as a base, and all the fluctuations of the gaps have been based on that. I don't think for Covishield ever there was a 28 days. You might have made a little misunderstanding. Something is a problem. It was always 6 weeks to begin with, then it becomes 8 weeks, and now it is 12 to 16 weeks. Ideally, because it was a dire emergency, and during the course of time, people have been found that some people developed the immune response within 2 weeks as it was expected, while in some people, it did not develop immune response in 2 weeks, and also the sufficient load of antibodies whether they will decline or not decline, people were worried about. That's why initially for Covaxin, it was a 28 to 30 days. And for Covishield, it was a 45 days, if I'm correct. And then what happened? The antibody stability has been found, at least in the case of Covishield, for up to 3 to 4 months. And that is why I think now the new guideline has come. The gap of booster has been moved from 6 weeks to anywhere 12 to 16 weeks, Javin sir. Okay, thank you very much.
Thank you, sir.
Thank you, sir.
Now we are having questions in chat box. The first question is, what are the different parameters which determines the efficiency and effectiveness of the vaccine? Sir?
Efficiency and effectiveness, basically what usually you're supposed to look typically, like in the field of microbial infections, like when you are looking for, there's something called a diagnostic titer. So once you vaccinate an individual, whether it is a recombinant vaccine, acellular vaccine, a portional vaccine, or RNA, DNA based vaccine, or the whole organism as a live or attenuated killed vaccine. In that case, what is expected is that the immune response is to be developed, and immune response should be developed in a significant amount. That is what the diagnostic titer, what is called. And the immune response should be able to neutralize efficiently the pathogenic agent against which the vaccine was developed. So when these two things are effectively happening in an individual or a large number of individuals, then that vaccine is called effective.
Thank you, sir. Next question is, do mutation affects the efficiency of vaccination?
Well, that's an imaginary question, because this is all the news media and WhatsApp and social media has spread so much of it. There is a fear and there is also some comfort. Largely fear, because it's a human tendency to read and look at the remotest bad possibility, because it's a truth that 90% patients did not even require hospitalization. And only around 2 to 3% patients have required the oxygen, whatever, whatnot. But the whole news, all the newspapers, news media, and WhatsApp is talking about the death, talking about the failure, but there has been a 95 to 97% success. So I think that is the one thing. But the second thing is that mutations, which mutation? Why I'm saying all this? Because which mutations we are referring to? That's the question. The mutation that we know from the RT-PCR are for the envelope gene and the N gene. Possibly, these mutations may not have a role in evading the immune response by the vaccine, but there is a possibility that vaccinated population at the global level has been found to be infected with the different strains of the COVID. So there is a possibility that immunity either is not sufficiently developed, that's why the reinfection, immunity declined after developing, that's why the second infection, or actually the strain is, the mutational strain is able to surpass the quality of immunity developed by the vaccine. All these three possibilities are so without experimental or evidences, it would be deleterious, poisonous to comment on this question.
Okay, sir. The next question is, some drugs are DNA manipulative drugs. So in country like India, which has vast variations in climatic and environmental conditions, is it possible that due to lack in preservation process that DNA manipulative drugs can give mutations like circumstances and can change its properties?
No, I think, are you talking about a vaccine or a drug? I think that's not very clear. But any drug that is being sold from the pharmacy has always been ensured to be kept at the appropriate temperature. And no matter whether in India or any part of the world, the person who is being treated has a 37-degree in the body temperature. So I don't think one needs to worry about the DNA targeting drugs are not properly acting in India and acting only in the first world country. No, it's not like that.
Okay, sir. Thank you, sir. The next question is, how long can a clinical trial of a vaccine take?
Yeah, that has already been told, like at least a year or two, it can go, because the mass of the populations are very huge, and you never know, because it's always a probability in the biology. You never have 100, you never have a 0. Everything is between 0 and 100%. So even if one day, one worst case comes up, after like crores of people without any complaint, all of a sudden new issue will start from that day. So we cannot really say, but normally 1 to 2
Well,
days.
Dr. Kharat, this is Samir Naik from Serum.
Yes,
If
sir.
you may please permit me
Yeah,
to, take this particular question on clinical trial. With your due permission, I would proceed.
please.
Yes, sir. Please continue.
So the clinical trial, again, of course, is a huge chapter, and there are several factors we need to consider for clinical trial and the design of clinical trial. The first of all is what is the objective of the clinical trial? One thing. Second thing, whether it is for the new molecule or the new drug or the new vaccine, whether it is for the already developed and clinical trial has been proved in some other countries, or uh, what is the population target. So these are the three important aspects that is covered uh, and govern the clinical trial design. And of course, like it depends on the number of population and the target population. Say for example, now just for an example, we did the rotavirus vaccine clinical trial, where I was, this was my one of the first projects. So we did this trial on approximately 15,000 infant of 6-week old to 9-week old. So the trial went for almost 3.5 years. This is a full-fledged efficacy trial, and it depends. Like if the target population is adult, if it is an adolescent age, or if it is for infants. So all these design could because for example, if it is for infant, we don't know that when the infant is coming. For adults, like that we know that these are the adults, we can appeal them. But for example, for infants, we have to really dependent on the pediatricians, the gynecological hospitals and other thing. So this is again challenging situations, and then we need to again convince the parents, all the DSMB board, etc. So of course, like roughly, like if you see, the clinical trial would take from maybe 1 year, 6 months, maybe to up to 4 years also. It depends on the trial design.
Okay. Thank you, sir. Next question is, which are the steps used to discover the drug for multi-drug resistant organisms, which are creating problems in today's world? What will be the strategy to treat the multi-drug resistant organisms?
So in this case, one has to identify a drug molecule against a particular pathogen. Let's say particular pathogen, there is like a group of pathogen called escape pathogen, let's say. It is the hospital community acquired infectious group, which covers 6 different pathogens, such as Enterococcus, then Staphylococcus, then Klebsiella, then Acinetobacter, then Pseudomonas, and then Enterobacter. So if the prevalence of these pathogens is more, and what are the challenges particularly in these pathogens? What are the commonly used antibiotics, third generation cephalosporins, and the combinations with beta-lactamase inhibitors, etc. For example, certain common aminoglycosides which have been used very generously for treating the gram-negative infections. Identify that and identify the strains which are highly resistant, not just a resistant, but they have a very high level of minimum inhibitory concentrations. And these are the candidate organisms are to be used for the search of antimicrobial agents, either from the chemical library or the chemical synthesis or the natural compounds, which are either from the natural sources, water, the soil, the medicinal plant, or any other sources, animal, plant origin. And one has to perform the studies first into the laboratory with these strains, not the laboratory strains, basically the pathogenic strains, which are highly drug resistant. Then try them out with many such kind of clinical isolates. And then one can move from the preclinical stages into the animal model. Like animal, you have to challenge the animal with these pathogens, then neutrogenize those animals, and then try to again give the treatment of the standard drugs. And in comparison with the standard drug, this molecule, whether it is efficiently clearing or not efficiently clearing, what are the immunological changes that in animal takes place, what are the toxic effects that molecule shows into the animal, that also has to be taken into consideration. How this drug is being retained, for how long it remains into the body, and how quickly, efficiently one can remove the traces of that compound from the body. Otherwise, what will happen, the animal will be free from the pathogen, but the compound itself remains into the body for a longer period of time. And then it may impart some kind of anomalies, which will have a longer cost to be paid. So these are the different steps to be taken at the preclinical stages. And once the animal stage has been approved, then the molecule is called a druggable molecule, which will go for the approval of the clinical trials.
Okay, thank you, sir. I want to ask the question related with multi-drug resistant organism. Is it really feasible to use the FAST therapy for human infections, which are caused by multi-drug resistant bacteria? Because
it's a very interesting and what is called like dreaming kind of a possibility that whole world is looking after. But the biggest problem of the phages is that they're highly host specific. And I would like to bring the story of S. Benzer's work, which I'm sure all microbiology students are studying. E. coli K12 strain and E. coli B strain. The same phage can't infect B2C or B2K. So if that kind of specificity is there for the phages, and the clinical strains are very different from each other, so there is always a possibility some erratic pattern of the effect can be expected with the phage therapy. Another thing is the milieu, the human body, will that be able to attract a kind of a phage receptor interactions with the bacterium and efficiently clear. But at this moment, it's a very beautiful and attractive hypothesis, and a lot of laboratories around the globe are considering phage therapy as a very promising technology, especially against the multi-drug or the extensively drug resistance pathogens.
May I ask a question?
Yes, sir. Yes, ma'am.
I have two questions. My first question is for Dr. Naik, sir. Sir, we all know that Covishield is a type of a vaccine where you have to calculate risk-benefit ratios and then decide you have to administer it or not. So on that line, many European countries have limited the AstraZeneca vaccine to the elderly people and recommended vaccines other than AstraZeneca for those below 30 years, especially in women. Now in India, I'm fully aware that we do not have that luxury, but as a vaccine maker, do you think that we should look more into it, or the serum has come up to any sort of explanation regarding these adverse events?
Can you just please repeat the question? So just missed your first part because of the internet connection.
Sir, many European countries have limited the AstraZeneca vaccine shot to elderly people due to the adverse events. So they have recommended it below 30 years of age, especially in women. So India do not have that luxury to do so right now. So do you think we should look more into it, or has serum has come up to any sort of explanation regarding the adverse events?
I would, slightly use the appropriate word rather than the luxury that India has. India does not have any luxury as such. Because it is not per se luxury, it is based upon the rules and regulations and the regulatory setup. Why the adverse event, particularly blood clots and other thing, was observed in Western world is again a matter of investigation. However, as a part of a manufacturer, I would just tell you that we have supplied and so far whatever the population of India has been vaccinated, almost amongst the individuals that are vaccinated, 80% of them have received Covishield vaccine, and we have reported none of the any adverse event which are life-threatening. Very few mild adverse events which are solicited has been reported. Of course, at this point of time, I do not have exact data in front of me about the solicited and unsolicited AEFI that were observed, but it is be assured that the Covishield vaccine is safe throughout the world. There are certain incidences has been reported in some part of the world, so you cannot generalize from one incidence for the entire world, because there are certain differences that you would observe with respect to the geographic location, the nutritional status, immunological status of the individuals who have received the vaccine. So it is not the luxury that I would, slightly, what I can say, will not use this particular word that India does not have any luxury. Yes, India has a full right, if at all any adverse events have been observed with any of the vaccine, to stop the use of vaccine. So far, what we have observed is there are no such kind of serious events or even blood clots that has been observed. As I already informed you that 80% of vaccinated population has already received Covishield, and the results are encouraging.
Yes, sir. Thank you very much.
Thank
My
you, sir.
second, shall I ask my second question now?
Yes, yes, you can continue.
My second.
Yes, please.
Hello, ma'am.
Sorry, ma'am.
Yes.
My second question is for Kharat, sir, Dr. Kharat, sir. So the United States and other developed countries have supported the lifting of patent protections on COVID vaccines so that poor countries like India and South Africa can get more vaccine dosages. So do you think just lifting such vaccine patent protections is enough, or will that solve the problem of vaccine deficiency?
I don't think patent protection would be sufficient availing. Actually, the vaccine is the need of the hour. I think that second part has already been discussed over medias in last 2 weeks. That is a big topic. So just waiving partially will not suffice the need of the third world country or the high density population around the globe. So that's not sufficient for sure.
So thank you.
Thank you, sir. Now, the question from Pradnya Mitke. How do we benefit from multi-layered tablets in pharmaceutical industries, and what are their advantages? Am I audible?
Yes, you're audible.
Okay, yes.
So what is the question?
And the second question is, should researcher of traditional, yes. How do we benefit from multi-layered tablets in pharmaceutical industry, and what are their advantages? She is asking about multi-layered tablets.
What is multi-layer tablets? What does it mean here?
Question is not cleared, I think.
Hello, I
Yes.
want to ask one question.
Yes.
Whenever any vaccine is
Yes, you can continue.
Hello.
Yes.
Whenever any vaccine is being developed, how the route of administration is decided, and how much it affect the immunogenicity and efficacy of the vaccine.
See, most of the vaccines are administered parenterally. That means they are injected either intramuscularly or intradermally, and very few are subcutaneously. So these are the preferred route. And if we look at the recognition of the antigen given by intradermal route or intramuscular route, the chances of antigen presentation uh, with or without adjuvant is good, so that you get a good immune response. And there are some vaccines which are given orally depending upon the type of an antigen. Say, for example, now for polio vaccine, it is oral polio vaccine. The purpose of developing the oral polio vaccine or oral rotavirus vaccine is because these are the infections which are caused by the enteric pathogen, and the immunity needs to be developed at the enteric site. Now, similarly, like in recent years, from 2010, a new concept has come is intranasal injection. So these are all the mucosal surfaces, like say for example, the oral vaccine, which again goes to the intestinal mucosal surface. The nasal vaccine is also going to the nasal mucosal surfaces, and a type of immune response that is required for a specific pathogen. Now, say for example, those pathogens which are respiratory pathogen, they would require IgG and IgA type of response. So the site of inoculation is either parenteral or the mucosal where it is intranasal. Depending upon the results of the experiment that is performed in the group during the clinical trial, where you have evaluated the vaccine immunogenicity with respect to the people who are inoculated parenterally as well as the intranasally, then you compare both of them. After the comparison of those results, then you decide which route would be better. So that is how then it is decided.
Thank you, sir.
Thank you,
The
sir.
next question is, many patients found COVID positive after taking 2 doses of vaccine. How it happens, as we know, after taking second dose. As we know, the memory
The reason is very simple:
cells get
negligence.
activated after taking the second dose.
See,
Yes.
what is important is that you are vaccinated. You took the vaccine. It may not necessary that people in the community, everyone is vaccinated. So it is a fact that the people around you are carrier or they are infected. So even if you are vaccinated, there is a likely that you get an infection. But of course, like those who are, Miss Dilare from TY BSc, probably she has asked this question. So even if the patient has been infected after the 2 dose, uh, it is sure, and what we have observed is that whatever the infection these people are having is very mild infection. So the vaccinated individual showed the infection, but it's a very mild infection, and the hospitalization is not required under this situation. On the contrary, if you look at the non-vaccinated population, the complications are there. Of course, like on the other side, from a common man's perspective, let me tell you that uh, it is the perception of common individuals that once you are vaccinated, that means nothing is going to happen to you. Well, that is not the case. This is a very wrong perception in the community that we observe people once you take vaccine, people are removing the mask and throwing the mask in dustbin, and they say that no, nothing is going to happen to me because now I'm vaccinated. And to a large extent, I would also say that please read the reports from the authentic sources. Believe all the news from the authentic sources. Like for example, if you are getting the information from ICMR Directorate or WHO, which are the prescribed sites. So about the vaccine efficacy as well as the immunogenicity information that you get it from the authentic sources. Uh, the chances of infection are there for even those who are vaccinated, because the vaccine has the efficacy up to 72%. But when we say that the vaccine has a 72% efficacy, so that means that if 100 people were vaccinated, out of 100, 72% people or 72 individuals got a protection. So when we say that, what about the remaining 25%? So they will get infection. But when you are going, increasing the immunization day by day, so you are increasing the number of vaccinated people, and the unvaccinated people will be very less. So amongst those population, we are not vaccinated, they are likely to develop the disease. And of course, within the vaccinated population, if you do not follow still the rules and regulation by wearing the mask, not coming in contact with the COVID infected individuals, following the good sanitization practices will help you to stay away from the COVID infection.
Thank you, sir.
Hello, sir.
Next question.
Yes,
Yes, sir.
please.
Yes, sir, you can ask.
Sir, as far as number of population in particular Maharashtra, as well as the vaccination, almost we have crossed 2 crore. So whether it is necessary that instead of giving second dose, we have to give first dose to maximum population, so that before third wave, whatever that proposed to say in September and October, whether we have to cover the maximum population.
No. Well, that is again.
May I have your attention, please?
Hello.
Wait.
Okay, sir. Okay, sir.
I'm sorry, I'm sitting in the conference room and all the buildings has the alarm systems. As you can, probably might have heard, it's an alarm system being activated because of some incidents probably that has happened in the building. Don't worry about it. It's a routine procedure.
No problem, sir.
So it's because of the noise I had to mute myself. Well, coming back to Dr. Pramod's question, I hope I take it correctly. I think, of course, this is our personal view that it is not a wise strategy to vaccinate everyone with a single dose and then you go for a second dose. Well, that is how the vaccine will not work. Vaccine has got a specific dose regime that once you receive the first shot of the vaccine, at least it has to be given after 4 to 6 weeks or 6 to 8 weeks as per the guideline given by the DCGI and the data that has been submitted. So the 2-dose regime with 6 weeks apart has to be followed to generate an effective immune response, so that you can have a kind of a protection and generation of a good immune response. Now, under this hypothetical condition, what you were saying, that suppose if you inoculate everyone with a single dose, it will take at least 6 months to maybe 8 months to inoculate everyone. And then the second dose will be given after 8 months. Then we do not know at this point of time whether it would be a wise strategy to give the second dose after 8 months, so that people would develop an effective immune response. At this point of time, the data suggests that this is not the way the vaccine is working. The vaccine is working with the 2-dose regime, and maybe we don't know whether we would require a third dose or not. We don't know at this point of time.
Samir sir,
Can I
Samir
add
sir.
into this one? Can I add into this one?
Yes, please.
There has been already so much of report, particularly with the COVID antibody. A lot of people have been found to recover and decline dramatically the antibodies within 3 months to 4 months span of time. That's a point number A. Point number B is with the SARS previously 2002, 2003 also, and with large number of flus, it is up to the 6 month, possibly anywhere between 3 to 6 months time, these antibodies are likely to decline and they don't have a very strong memory response like against the bacterial immunity is there. So that is also a reason if you extend the period of second dose by some mean, those all candidates will become like a first dose rather than becoming a booster second dose. So it's important to keep the second dose on a priority rather than hurriedly going for the first dose to all population. At least you would be fully vaccinating and boosting the immunity among those first shot received individuals and ensuring that the proper vaccination is done. That is what is the case with the COVID and all or the SARS viruses.
Sir, in this connection, I have one question. I have one question, sir, in this connection. How Sputnik 1 dose is enough? You were talking about Covishield. Russia.
The thing is that Sputnik has made use of 2 different vectors. So presently, there are like lot of speculations and scientific theories going on. One of the adeno-associated virus-based vaccine, Sputnik, is the most effective at this moment, considered to be. And there is also Moderna that is also claiming to be a single dose. Even now, the Virapin is also coming up with the similar kind of claim from the Zydus Cadila. So it's not surprising. It's not surprising, but the time will tell. Time will tell, because based on the earlier pre-existing literature, whatever I have told you the case. But we'll have to see whether these people will require to take or repeat the dose of vaccine. It has already been there with the cold or seasonal flu. Every spring beginning, one has to take the vaccine. One has to. So it's not even a 1 year. This spring to the next spring, you lose the immunity or you lose the memory. So you'll have to re-vaccinate. It has already been established. It has already been done. Probably the Sputnik will give you the immunity for 6 months or probably a little bit, but that one has to. There is no sufficient amount of literature available, sir. It is not yet available. We are all talking about emergency situation.
Okay, thank you, sir. Thank you.
And second important thing is that we are talking based upon the present data.
Yeah.
We don't know, very limited data is available, particularly for Sputnik. While for the others vaccine, you have very good amount of data has been available. But of course, will be very interesting to see how Sputnik vaccine will behave in the population. Yeah. And what we are looking at this point of time is only the clinical data, not actually the market data.
Exactly. There are 2 things, Gorpade sir. The double triple mutant that has been discussed presently is in India, and now Sputnik is being challenged against those strains. So that is a one thing. And how the genome of the genetic background of Indian population will also respond, that we have to compare in the light of Russian population. Okay. So it's a very preliminary stage to come to conclusion.
Okay, thank you, sir. Thank you.
Thank you, sir. Now, next question is from
I would like to ask one question.
NS Joshi ma'am. It is advisable to take Covishield to immunocompromised patients.
The answer is no.
Okay, thank you, sir.
Ma'am, I would like to
yes, you can. Yes.
My question is for Naik sir. Sir, there has been speculation about vaccine being ineffective for mutations that are arising in coronavirus strain, that double mutant and all. So I wanted to know how when the vaccine is developed or prepared, what strategies undertaken or studies done so as to ensure that the vaccine candidate can be effective towards more mutations, because mutations certainly take place in virus. It is a natural phenomenon. So there is possibility that even during the production phase also the virus can go mutate. So what different studies or strategies are undertaken to ensure that vaccine is effective towards these probable mutations?
Well, there are a couple of 2 or 3 questions that you have asked in one particular question. First of all, being a manufacturer and one of the person involved in this particular vaccine development, at this point of time, I think it is too premature to say that the vaccine is not effective against the mutant, because the data is still being generated. So I would not comment on that part, whether it will yes or no from this point of time. We have to wait for some time, because the mutant has arrived in India in October 2020 onward. The vaccine development took place earlier than that. So we are still, receiving the data and the effectiveness study, which is still ongoing, particularly against the mutant and other things. So it is extremely premature at this point of time to state about this. And of course, it will be like duly communicated through proper channel from our institute to the VCGI, as well as to maybe the common person. So this question I would answer at this point of time with this, that it's premature to comment on this aspect. The second part, when you said about the development, so when you develop a vaccine, and suppose now you imagine that in year December 2020, when the Wuhan virus was first officially been declared by the China that this is an infection that is being getting. In January, it is coming to different parts of the world. February, it is becoming pandemic. In March, we have gotten lockdown. That the chronology happened. So when all of us, including Serum or maybe Oxford, AstraZeneca, Moderna, Pfizer, when everyone started working on this particular Wuhan strain, the first sequence of Wuhan virus spike protein was available in January 2020. At that point of time, no one knew how the virus is going to mutate. And it is very unpredictable for viruses that how the mutation will take place, because the virus is getting passaged or replicated in different individuals among different localities, different geographical region, different community and races. So it's very unpredictable. But what was important at that point of time is to develop an effective vaccine against the strain which was reported. Yeah. Now, as we are learning from October 2020 onward that we are getting UK variant, South African variant, Indian variants. So of course, like the strategies are ongoing, and this particular vaccine, including Moderna, Pfizer, everyone who had developed this vaccine was against the original Wuhan strain of coronavirus. And subsequently, as we are learning different mutations, basically the strategies are being developed by different manufacturer and the research agencies to develop an effective vaccine against even the variants, and the efficacy of those vaccines are also being monitored. So this is the part second. So I hope this has answered your question. If you have any.
Sir, I have one question.
Yes.
Naik, sir.
Yes, please.
I have taken 2 shots of Covishield.
Yes, ma'am.
Yes, I have taken 2 shots of Covishield, and I'm afraid whether my antibodies will decline when there will be third wave. So
Madam, yes, with due respect, madam, and I know Dr. Wadikar, I believe, correct? Sanyogita, madam?
Yes, yes, sir.
I think before I answer this question, just on personal note, I have read your books when I was in first year of BSc on microbiology.
Yes, thank you.
Some other day, like it's coincident that we met. It's a very good book. And one of your student is my very good friend and colleague. He's from your university, Jagdish. We work together, Dr. Jagdish Jane. So we have heard many things about you and Dr. Wadikar.
Thank you very much, sir.
So as soon as your name has come onto the screen particular, so I thought took this opportunity that it's a pleasure, and the work that you did at your university is highly appreciable.
Thank you very much.
Yeah. Coming back to your question about whether your antibodies will decline and the third wave, I would say that we will cross the bridge when we will reach at the bridge. Third wave is imaginary at this point of time. We don't know whether really third wave is coming. And of course, it has been seen that immunologically, the weaning of antibody titer is observed even after vaccination to many people. So still, when we developed this particular vaccine, we never knew that whether we would require one dose, whether we would require two doses, whether the two-dose regime would be effective, and what was more important is to see the longevity of the antibodies and the longevity of the T-cell immune response.
Yeah.
And of course, this has triggered, one of the questions that has asked earlier to me regarding this particular virus and mutations. Like as we are learning about the mutations and we don't know how many mutations would come. And it is very intriguing as a virologist to understand that coronavirus per se is known to not so mutagenic. A very less mutations has been observed in SARS mutation and other things, but this particular strain is mutated very fast within the shortest possible time. Of course, viruses are known to have the mutations in different antigenic sites. It is surprising that coronavirus has mutated to such an extent so that it has formed different variants. Of course, we are still learning many things about the mutations, the different spike protein mutations and other mutations. But coming back to your question is, we really don't know what is the stage of third wave and what would be the immune status. But definitely, if you are vaccinated with any of the vaccine, whether it is Covishield, Covaxin, or any other thing, take it from us that there is a protection against the all existing strains, and definitely the severity of the infection has significantly reduced to certain extent that at least what we have observed with our very few limited examples, that those individuals who are vaccinated and got an infection, their chest is very clean and clear. Their RT, HRCT values are very less. They do not require hospitalization. They did not require any oxygen supply or any other thing. And forget about the ventilation.
Okay, so thank you. But whether the study has done on half-life period of the antibodies?
Madam, that's what I said, that it is extremely premature at this point of time. The studies are still ongoing. The people are getting vaccinated. It is from January 16, 2021, the vaccination program has started. It is only now we are in the 4th or 5th month after the vaccination. The people are being getting monitored, followed up. And what is more important is that under this exceptional condition, it is again difficult for government agencies, the clinical trial people, to follow the people who have been vaccinated. So there are a lot of difficulties, but of course that is not an excuse. The work is ongoing. The data will going to come very soon, and then we will come to know what will be the status of immunological memory among those individuals who have received the vaccine.
Thank you. Thank you very much.
Thank you.
Thank you, sir. In chat box, there are some questions related to DRDO drugs, to DG drugs. Can you elaborate about this consideration or about these drugs, that is its properties or its efficiency?
So what is the question?
The question is about the DRDO drug, that is 2DG medicine. So they want to ask about its properties or its action of mechanism or its efficiency.
Okay, since it's an academic mock, basically glucose is being processed or metabolized to produce glucose 6-phosphate, then into the fructokinase to fructose 6-phosphate. Now, T-deoxyglucose is a substrate analog. It can produce 2-deoxyglucose can produce glucose 6-phosphate, but it cannot convert into the fructokinase, cannot use 2-deoxygenated glucose 6-phosphate as a substrate to produce fructose 6-phosphate. So due to lack of fructose 6-phosphate, it will accumulate 2-deoxyglucose 6-phosphate in the cells, and as a result of which it will hamper basically the ATP generation, which is required and a source of a carbon for the proper replication of the virus. Now this is being implicated. Now the time will tell. Also, T-deoxyglucose is not given as a standalone. It is also referred during the clinical phase 3 only. Interesting point is that almost 2 decades before, the same molecule was tested for the anti-cancer property. So there is a lot that we have to learn about the number of viruses. However, at least in my opinion, if it is given during the inflammatory response, then the challenge of therapy is not about a virus replication, but also controlling the cytokine storm and the multiple systemic dysfunction of the in the patient. So that I don't know whether the 2-deoxyglucose will add into the problem or will help recovering, but in the initial stages, mild infections to moderate infections certainly are likely to get some help with the 2-DG, and that is also along with the other therapeutics such as your ivermectin, deoxycholine, and other like azithromycin. So that is what is recommended. But again, it is under the test, and time will tell its efficacy. It's a substrate analog competitive inhibitor.
Okay, thank you, sir.
Sir, I will
My
add.
next. Yes, sir, you can ask, sir.
Khara, sir, I will add in that it's the cheat to the cheater.
Yeah, it is. It is. That's what substrate analog and competitive inhibitor is. That it is a bad substrate. Yeah, so it won't be able to produce 2-deoxygenated fructose 6-phosphate will not be produced. So it will not enter. It will use one ATP, but it will not produce the another ATPs into it. So it's a bad substrate.
Okay, thank you, sir. My next question is, which are the current application of nanotechnology in the pharmaceutical product formation or formation? That is, nanoparticles with antiviral activities or antibacterial activities, can they use or are in use in pharmaceutical formation?
Well, yes, please, sir.
No, go ahead. Nanoparticles have already been used in lots of cosmetics, particularly. Antimicrobial, antiviral properties are also implicated, but it has to wait for some more further research in order to validate in the clinical trial for its efficacy and the safety. So it's still promising, but not ruled out at this stage or confirmed also.
Thank you, sir. Next question is about the blood group susceptibility. That is, according to CSR, people with AB and B
I read that question.
blood groups are more susceptible to COVID-19. What is your opinion, sir?
No, there's nothing like, there is no opinion on this question, because this is, I read this question. This is a public perception, actually. If you look at the people who have been infected, it would be statistically insignificant or may not be a very significant difference, it would mean that A blood group, O blood group don't get infected with the COVID. So that's not true. That's not true. Every blood group
Participants, you can unmute yourself and ask the questions. Thank you, sir.
I have a question. Vaccines which are adenovirus vector vaccines. So if we get the first dose, our body will produce antibodies against coronavirus spike proteins. Does it produce antibodies against adenovirus as well? And if yes, then will the vaccine efficacy will decline after repeated vaccine use?
Vectors are not used as a whole immunogenic part. It is being compromised, so that won't be the case. That is one of the side effects expected, and as of now, already Naik has mentioned that there has not been report in Indian subjects with the Covishield also. So at this moment, the benefit of generating immunity seems to be more prevalent than the consequence that is being questioned out here. So while using this defective viral part as a vector, the care has been taken.
Okay, sir. Thank you.
Thank you, sir.
Ma'am, if there are no questions, can you sum up? Because it is 35.
Yeah, please. Thank you.
Shinde madam, over to you, ma'am.
Yes, sir. Yes, sir.
Proceed.
Yes, sir. Now I would like to request Dr. V.M. Ghorpade, sir, to conclude the session.
Good evening, everybody. It was a nice session. Dr. Samir Naik, sir, and Kharas, sir. And good evening to everybody. I am Dr. Vilasrao Ghorpade. I am here to present the vote of the thanks. I would like to thank Dr. Samir Naik, sir, and Dr. Arun Kharas, sir, enlightening with their knowledge. Today, the panel discussion with full of knowledge and interesting facts about the vaccine development. It gives me deep insight into the topic and reveals some interesting facts. Once again, I thank you, Kharas sir, and thank you, Samir sir. I would also thanks to our principal, Dr. B.T. Jadhav sir, for giving the permission to organize such a YCIS hackathon. At last, I also thanks to both of you because of besides your busy schedule, you have made the event successfully. Once again, I thanks to the participants and organizing committee and OIT department for the technical assistance and teachers and the students to make this event very successfully. Thank you. By kind permission of the organizing committee, I declare that this event is over. Thank you. Thank you.
Thank you.
Thank you, sir.
Thank you, sir. Thank you.
Thank you. Thank you so much. Thank you.
Naik sir, Kharas sir.
Thank you.
Thank you so much for attending the session.
Thank you.
All the
Thank you.